Unraveling the Genetic Threads of Dyslexia, a landmark study. A groundbreaking genetic study has shed new light on the complex world of dyslexia, a neurodevelopmental condition affecting millions worldwide. Researchers from prestigious institutions, including the University of Edinburgh and the Max Planck Institute for Psycholinguistics, have embarked on a journey to decipher the genetic mysteries behind this learning difference. Dyslexia, a condition often accompanied by persistent reading and writing challenges, affects a significant portion of the global population, estimated to be between 5 and 10%. The study, published in Translational Psychiatry, delves into the genetic factors that contribute to this widespread issue, offering a more comprehensive understanding of dyslexia's origins. The genetic heritage of dyslexia. Previous studies have hinted at the hereditary nature of dyslexia, but the specific genetic variants responsible remained elusive. Haley Mountford, a research fellow at the University of Edinburgh School of Psychology, explains, "This research was motivated by the long-standing challenge of identifying the genetic basis of dyslexia, a common and often inherited learning difference. The study's innovative approach involved analyzing vast genetic datasets, combining detailed reading ability test data from the GenLang Consortium with genetic information from over 50,000 individuals diagnosed with dyslexia through 23andMe. This meta-analysis identified 13 new genetic loci linked to dyslexia, providing crucial insights into the early brain development processes affected by this condition." Expanding horizons in dyslexia research. Despite significant progress, dyslexia research still lags behind studies of conditions like autism and ADHD. Mountford highlights the importance of this work, stating, "Research into dyslexia is still far behind that of autism or attention deficit hyperactivity disorder, ADHD, and the biological mechanisms remained unclear." The study's primary objectives were twofold: uncovering new genes associated with dyslexia and gaining a deeper understanding of the biological basis of reading ability variations. Additionally, researchers explored the potential of genetic scores, known as polygenic indices, to predict reading difficulties. Unraveling the genetic architecture. The study's findings are remarkable, identifying 80 regions associated with dyslexia, including 36 previously unreported significant regions. Among these, 13 were entirely novel, significantly expanding our knowledge of the genetic architecture of reading-related traits. Interestingly, many of these genes are active in brain regions still developing during early life, supporting neural signaling and synapse formation. Predicting reading difficulties and evolutionary insights. The polygenic index developed by the researchers demonstrated a modest yet significant ability to predict reading ability, explaining up to 4.7% of the variance. This finding paves the way for potential early identification of reading difficulties. Furthermore, the study revealed no evidence of recent evolutionary selection for or against dyslexia-associated genes, suggesting that dyslexia has not been significantly influenced by societal changes over the past 15,000 years in Northern Europe. This landmark study marks a significant advancement in our understanding of dyslexia, offering hope for improved early identification and support for individuals affected by this condition.